Hello everyone. In this video, you will learn about various transport mechanism across membrane. Cell membrane is semi-permeable, where it allows only certain substance to pass through it easily, while some substance require transport protein to pass through. Two main types of transport mechanism are passive transport and active transport. Passive transport is the transport of substances across membrane, down the concentration gradient, or from high concentration to lower concentration. ADP is not required. Meanwhile, active transport is the transport of substances across membrane, against the concentration gradient, or from low concentration to higher concentration. ADP is required. Three types of passive transport are simple diffusion, facilitated diffusion, and osmosis. While two types of active transport is sodium-potassium pump and bulk transport. Bulk transport occur by two mechanisms: endocytosis and exocytosis. In simple diffusion, diffusion of substances from a region of high concentration to lower concentration does not require transport protein. This is due to the characteristics of the substance, which are small size and hydrophobic, or lipid-soluble, or nonpolar, or uncharged molecule. Example of hydrophobic or lipid-soluble molecules are testosterone, estrogen, and vitamin A, D, E, K. Example of nonpolar, or uncharged molecules are oxygen and carbon dioxide. In facilitated diffusion, diffusion of substances across membrane from region of high concentration to lower concentration require transport protein, such as channel protein and carrier protein. Channel protein has no binding site. It transports charged or ions such as sodium ion, calcium ion, chloride ion, bicarbonate ion, and potassium ion. Meanwhile, carrier protein has binding site for specific molecules. It transport polar or hydrophilic, relatively large molecules, such as glucose and amino acid. Osmosis is a transport of water molecules across membrane, from a region of high water potential, or high water concentration, to a region of lower water potential, or low water concentration. Osmosis is the net movement of water molecules across semi-permeable membrane, from a region of high water potential to a region of low water potential, without energy requirement. The more the concentrated a solution, the lower its water potential. For example, if we compare a cup coffee and a glass of water, coffee has lower water potential compared to water, because coffee is more concentrated. By comparing the water potential, we can determine which region has the highest water potential, and the lowest water potential. We also can determine the direction of water diffusion between different regions. In this diagram there are three regions with different water potential, that are separated by semi-permeable membrane. Region A is a pure water, with 0 kPa water potential. Region B is dilute solution, with negative 200 kPa water potential. Region C has concentrated solution, with negative 500 kPa water potential. Region A has the highest water potential. Region B has lower water potential than A, but higher than C. While Region C has the lowest water potential. Because of water diffuse from a region of high water potential to lower water potential, water from Region A will diffuse into Region B and C, while water from Region B diffuse into Region C. Due to the different potential of cells and its surrounding, we can calculate water potential of cells and the solution surround it. Water potential can be calculated by using this formula. Symbol of water potential is psi. Water potential equal to pressure potential plus solute potential. For example, calculate the water potential of a plant cell, with a pressure potential of 0.8 megapascal, and an osmotic potential, or solute potential of -1.6 megapascal. Calculate water potential. From the question, the pressure potential is 0.8 megapascal, while the solute potential is -1.6 megapascal. So, we can calculate the water potential by using the formula. The water potential for the cell is -0.8 megapascal. The water potential of a cell is always negative value, because cell contains solutes. Solutes make a cell more concentrated, therefore, water potential become negative value. The solution surrounding the cells will determine whether a cell gain or loss water. The ability of a solution that cause a cell to gain or loss water is known as tonicity. There are three types of tonicity, which are hypotonic, isotonic, and hypertonic. Hypotonic solution is a solution with higher water potential or lower concentration of solute than the cytoplasm of cell. If an animal cell and plant cell was put in a hypotonic solution, water will diffuse into the cell, causing the animal cell to become lysed, while the plant cell becomes turgid, which is normal for plant. Isotonic solution is a solution with equal concentration of solute and water between the solution and the cytoplasm of cell. If an animal cell and plant cell was put in an isotonic solution, water will diffuse into and out of cell at equal rate. The animal cell remains in normal condition, while the plant cell will becomes flaccid. Hypertonic solution is a solution with lower water potential or higher concentration of solute than the cytoplasm of cell. If an animal cell and plant cell was put in a hypertonic solution, water will diffuse out of cell, causing the animal cell to shriveled, while the plant cell becomes plasmolysed. In this picture, the region A and B is separated by semi-permeable membrane. Region A is more dilute than region B. Therefore, region A has high water potential, and region B has low water potential. Because of region A has high water potential than region B, solution in region A is hypotonic solution. The concentration of water is higher than concentration of solute, while solution in region B is hypertonic solution. The concentration of solute is higher than concentration of water. The semi-permeable membrane is permeable to water, but impermeable to solute. So, water can cross the semi-permeable membrane, but solute cannot. Therefore, water molecules from region A will diffuse into region B, across the semi-permeable membrane from the region of high water potential to lower water potential, until both sides reach equal water potential. This will result an isotonic solution. Both sides has equal water potential. Therefore, water molecules moves between region A and B at the same rate. Unlike passive transport, active transport will transport substances across membrane down the concentration gradient, where ADP is needed. Sodium-potassium pump is an example for active transport. Sodium-potassium pump is a carrier protein that is used to maintain the concentration of sodium ion and potassium ion across membrane. 3 sodium ions will be pumped out of cell, and 2 potassium ions will be pumped into the cell. This process require energy or ADP. Firstly, in the intracellular, 3 sodium ions bind to binding site at sodium-potassium pump. ADP will bind to sodium-potassium pump and provide energy to the carrier protein. Sodium-potassium pump gain energy, and change its conformation or shape. The 3 sodium ions then will be pumped out of cell. 2 potassium ions from the extracellular will bind to binding site of sodium-potassium pump. Binding of potassium ions will trigger the sodium-potassium pump to return to its original conformation. And then, the 2 potassium ions will be pumped into the cell. A new cycle is ready to occur. Bulk transport is the movement of large molecules, for example, protein and polysaccharide, large particles, large amount of molecules, across the membrane. Bulk transport involved the formation of vesicles or vacuoles. This mechanism is an active transport, because this process requires ADP and substances move against the concentration gradient. Two types of bulk transport is endocytosis and exocytosis. Endocytosis is a process where a living cell taken up or engulfed in bulk. Meanwhile, exocytosis is a process of transporting materials or substances out of cell in bulk. Endocytosis divides into two types, which are phagocytosis and pinocytosis. Phagocytosis is a process where living cell taken up or engulfs solid particle or bacteria. Plasma membrane forms pseudopodium to engulf solid particle, and then pinched off. Food vacuole is formed. Lysosome then will fuses with the food vacuole. Hydrolytic enzyme in lysosome will digest the solid particle or bacteria. Materials is then absorbed into cytoplasm. Pinocytosis is a process where living cell taken up or engulf liquid particle or solutes. Plasma membrane invaginate to engulf solid particle, and pinched off. To form vesicle. The materials are then absorbed directly into the cytoplasm. Phagocytosis is known as cell eating, because the cell engulf large and solid particles. Meanwhile, pinocytosis is known as cell drinking, because the cell engulfs small and liquid particles. In exocytosis, vesicles or vacuoles contains materials from the cell, fuses with plasma membrane, and then, the contents in the vesicles or vacuoles are released to the outside of the cell. Example of contents in the vesicle or vacuoles are enzymes, hormones, waste product, mucus, etc. Well class, that's all about cell transport mechanisms. As a conclusion, passive transport is the movement of molecules across semi-permeable membrane. From a region of higher concentration to a region of lower concentration, or down the concentration gradient, without energy requirement. While active transport is the movement of molecules across semi-permeable membrane. From a region of lower concentration to a region of higher concentration, or against the concentration gradient, with energy requirement.